Alright, let's come back to the theoretical questions. You might remember that we had attempted this question earlier but had set aside some of its parts to cover later in this chapter, so let's tackle them now. If you look at figure 6.1, you'll see it shows a biological molecule with a double helix structure. The question is, name this molecule. The answer, of course, is deoxyribonucleic acid, commonly known as DNA. Next, they ask us to describe 3 features of the structure of this molecule. Let's think this through. Firstly, DNA has a double helix structure, which means it looks like a twisted ladder. It is made up of 2 strands. We call these 2 polynucleotide chains, coiled around each other. That's the first feature. Secondly, DNA is composed of elements such as carbon, hydrogen, oxygen, nitrogen, and phosphorus. These elements form both the sugar phosphate backbone and the nitrogenous bases of the molecule. And thirdly, DNA is built from smaller subunits called nucleotides. These nucleotides are arranged with complementary bases facing each other, forming the rungs of the ladder-like structure. Adenine pairs with thymine, and guanine pairs with cytosine. These complementary base pairs hold the 2 strands together. So these 3 points, the double helix shape with 2 strands, the key chemical elements, and the nucleotide subunits with complementary base pairing, are enough to answer the question fully and correctly according to our marking scheme. Then, let's move on to the next part of this question, which connects the cells chapter back to DNA so that we can revise and strengthen your understanding. The question tells us that both animal and bacterial cells contain DNA, and asks us how DNA controls cell function. But before that, it wants us to describe how the location and shape of DNA differ in these 2 types of cells. So we can note that in animal cells, DNA is located inside the nucleus, whereas in bacterial cells, the DNA lies freely in the cytoplasm, because animal cells are eukaryotic with a membrane-bound nucleus, while bacteria are prokaryotic and lack a true nucleus. As for the shape, DNA in animal cells is linear and double-stranded, organized into chromosomes, while bacterial DNA is usually a small circular double-stranded molecule. Then in part C, explaining how DNA controls cell function, we can say that DNA contains genes, which code for proteins or polypeptides. The sequence of bases in DNA determines the order of amino acids, and these proteins then control the structure and activities of the cell. Proteins carry out vital roles, for example as enzymes to speed up reactions, as structural components like collagen, as transport proteins like hemoglobin, as antibodies in the immune system, or even as hormones like insulin. So overall, by controlling which proteins are produced and in what order, DNA directs every process that happens inside the cell. Coming back to multiple choice questions, the question says that the student carried out 2 tests on a liquid. First, they added Benedict's solution to half of the liquid and heated it, which turned the solution red. Then they added Biuret reagent to the other half, and it turned mauve or purple. Let's break this down. Remember, Benedict's solution is used to test for reducing sugars like glucose, and the red precipitate is a positive result. The Biuret test is used to detect proteins, with a purple or mauve color indicating their presence. So from these results, we can conclude that the liquid contained both glucose, a reducing sugar, and proteins. That's why the correct answer is option D, which states that the liquid contains solutions of glucose and protein. Moving on to the next question, it asks about enzymes, which are involved in many reactions in living cells. Which statement describes the action of enzymes? Well, enzymes are biological catalysts. They increase the rate of biochemical reactions by lowering the activation energy required, and they are not used up in the reaction. So the correct answer here is option C, which says that enzymes increase the rate of chemical reactions. This ties directly back to our study of biological molecules, because enzymes themselves are proteins. That's a crucial point. When you see the word proteins highlighted in your notes or on a diagram, remember that enzymes are a vital class of proteins that carry out essential roles in the cell by speeding up life-sustaining reactions. Alright, everyone, let's turn to the next multiple choice question, which focuses on chemical tests for substances in urine. It mentions that certain health problems can be identified by checking for protein and glucose in the urine, and asks, which row shows the results for a person who has both protein and glucose present. Let's quickly remind ourselves of the key chemical tests involved. For glucose, we use Benedict's solution, and remember that this requires heating. A positive test result for reducing sugars like glucose causes Benedict's solution to change from its original blue color through green, then yellow, orange, and finally to brick red, depending on the concentration of glucose. For protein, we use the Biuret reagent. If protein is present, Biuret changes from blue to a mauve or purple color. There are, of course, other food tests you may have studied, such as the ethanol emulsion test for lipids, which goes cloudy or milky if lipids are present, and iodine solution, which turns blue-black with starch, but these are not relevant to this particular question. So, let's apply this to our scenario. If a person has both glucose and protein in their urine, what should we see? Benedict's solution should turn red, indicating the presence of glucose, while Biuret should turn mauve, showing the presence of protein. Now, looking at the answer options given, B matches exactly these results. Benedict's turning red, and Biuret turning mauve. Therefore, B is the correct answer.